All right, we're transitioning in the class now away from membranes, transporters, and signaling pathways, and we're going to move to talk about the next uh, biomolecular structure, and that is carbohydrates. And today we're going to be focusing really on sugars, the building blocks of longer carbohydrates. And so today, the material we're going to be covering is from chapter 7, and in particular, section 1. Okay, we're going to talk about monosaccharides and disaccharides, which are, monosaccharides are just single sugars. Disaccharides, we'll get into a little bit. Those are when you have two monosaccharides attached together. Okay. We're going to go through the structure of these sugars, a little bit of the nomenclature, and then we're going to talk about different isomers and some terms that will come up. Uh, one is enantiomers, and those, if you remember from organic chemistry, those are non-superimposable mirror images. Okay? And for sugars and amino acids as well, we refer to enantiomers as the D and the L form, and I'll explain really what that means, how you come to find what enantiomer a sugar is. Okay, and then there's another term that we'll use is epimer, and we'll explain that later. Um, we'll talk about the open forms of the sugars and also the ring forms. So sugars can exist in two different forms, okay, and they kind of have an equilibrium between the two. Okay. Glycosidic bonds, those are formed when you get two sugars or more sugars attaching together, bonding together to form chains. Okay, and then there's another type of structure designation for these, and that's known as anomers. And there's two forms, the alpha and beta anomer. Okay, and finally, we'll talk a little bit about what's a reducing sugar and what's not a reducing sugar. Okay, so starting out, defining the term carbohydrates. Okay, and that name, as it suggests, carbohydrates are molecules that are formed by carbon and what is really water. Okay, so you have carbohydrates take the form of C sub N, so a certain number of carbons in the chain, and every carbon, for every carbon there is two hydrogens and one water. Okay, so that they have the same N subscript there. So that's really why they call it carbohydrates. You have carbon and water. Okay, these are produced from plants, photosynthesis, and primarily plants. Okay, where they take carbon dioxide from the air and water, and they form them. Okay, these are very a wide range of molecules. They're the smallest carbohydrate we really consider a carbohydrate is glycerolaldehyde, which has 3 carbons, and its molecular weight's 90 grams per mole. And they can be as large as the carbohydrate amylopectin, which we'll talk about next lecture. Its molecular weight can be 200 million grams per mole, so like enormous molecules, and then uh, all the way in between those two, right? So structurally and size wise, these are a very diverse class of biomolecule. Right? They fulfill a variety of functions. One, primarily, probably the most obvious, is an energy source, also energy storage. Okay. They also provide structural integrity, so things like cell walls in bacteria cells or exoskeletons in certain animals and insects. Okay. They Another role that they they have is that we don't really talk a whole lot about in this class, but they do provide information, and especially on the outside of cells in cell-cell signaling, these carbohydrate signals, that's where they're involved. Okay, so they do have a pretty wide range of functions. Okay. They can be linked with proteins to form what are known as glycoproteins or proteoglycans. Okay, so they can also combine with proteins. Yeah. So let's look at some really very basic sugar, glycerolaldehyde. Okay, it's 3 carbons long. It has this general structure. Okay. You can also have a similar 3-carbon sugar, but notice that the carbonyl carbon, instead of being on the end, it's in the middle. Okay, and so this is actually known as dihydroxyacetone. So dihydroxyacetone, excuse me. These are the two types of sugars that we consider, and one is an aldose, glycerolaldehyde, and it has, when its ketone, excuse me, when its carbonyl group is on the end, this functional group is an aldehyde. Okay, so we call sugars that have the carbonyl on an end at the end of the chain aldoses. So this is an aldose. Glycerolaldehyde is an aldose. Dihydroxyacetone, when the carbonyl group is in an interior carbon, that forms a ketone functional group, and so we call this a ketose, right? So aldo and keto stand for aldehyde and ketone, and the ose, the OSE, means it's a sugar. Okay. We can also go a little bit further and say glyceraldehyde is an aldotriose or aldotriose. That means it's a 3-carbon long sugar. Okay, and likewise, dihydroxyacetone is a ketotriose. Right, it's a ketose with 3 carbons. Okay, and as we've mentioned before, OSE indicates that these are sugars, and like we've seen before in class, very similar when something has an ASE, an ACE on the end of it, it's because it's an enzyme, but OSE, that denotes that it's a sugar. Okay. So drawing sugars, a lot of times you'll see sugars drawn in what's known as a Fischer projection, and this is an example of Fischer projections for glycerolaldehyde. Okay. They don't show geometry very well. Okay, what this is a very simple way of depicting the sugar. Okay, what you should note that, right, it looks like the carbon here, and there's 4 bonds around it in sort of a square planar arrangement. But remember, carbon is tetrahedral, right? So this isn't showing geometry. So what you need to, when you look at a Fischer projection, what you need to remember is that these bonds are going to either be facing towards you or away from you. So in this projection, you can think of the horizontal bonds as being towards us, coming out of the screen, and the vertical bonds going into the screen. Okay, so if you wanted to show this in a perspective, what's known as perspective formula, which would be more of the 3-dimensional way of showing it, this is how that would look. Okay, and so glycerolaldehyde is probably a bad example for Fischer projections because there's only 3 carbons, right? If you have more carbons, these bonds would then start alternating, right? So if you had another carbon up here in the sugar, this bond is going out, into the and into the screen, the next bond coming off of this carbon would be coming out of the screen. Okay, so the bonds would alternate. Okay, the other thing I wanted to mention here, and this is where we're going to talk about enantiomers of sugars, the D and the L forms. They're all based on the direction of glycerolaldehyde, the OH group in glycerolaldehyde. Okay, so for glycerolaldehyde, when we draw it this way, when the OH group is on the right side, this was what we call the D enantiomer. When it's on the left side, this would be the L enantiomer. And you could kind of go through this if you looked at this carbon and then you use the hand rule to try to figure out which of these what enantiomer it is in terms of R and S, you could calculate that out. Okay. What else do I want to say about that? So in this class, the R and S designations we won't use. We're going to be using the D and the L. And again, it's a little bit, I guess, it's different because they're all, the D and the L are all going to be based on the glycerolaldehyde, the direction in the Fisher projection of the OH group. And I'll explain a little bit more about that. Okay. So enantiomers, just a reminder, enantiomers are stereoisomers that are non-superimposable mirror images. Okay. Sugars that contain many chiral centers, okay, so if you have a chain of 6 carbons, there's going to be actually quite a few chiral centers in that carbon, but only the one that is most distant from the carbonyl carbon is designated as D or L. Okay, and D is sort of the right-handed, and L stands for the left-handed. Okay. Okay, D and L isomers of a sugar are enantiomers. They're non-superimposable mirror images of each other. Okay. When you have these D and L enantiomers of a sugar, those molecules are going to have very, very similar properties. Okay, so things like water solubility are going to be the same. The thing that is going to be different is the way they rotate polar play. What am I trying to say? Polarized light. The way they rotate the polar light is going to be different. Okay? But their physical properties, other than that, are going to be identical. Okay. Most 6-carbon sugars, so hexoses, in living organisms are D enantiomers. There are some L forms, but primarily sugars are in the D enantiomer form in living organisms, which is a little bit different than amino acids. Remember, amino acids, in general, most amino acids in our bodies are the L enantiomer. And just casting our mind back to organic chemistry, if we use a ball and stick model now to depict glycerolaldehyde, right? If you have a mirror image of one form, okay, so this would be the D form. If you take the mirror image of that and you try to superimpose those on top of each other, it's not going to work, right? So you can line up the OH and the carbon together, but when you do that, you're going to have, let's try to do that. Okay, so you could line up the OH, the carbon, and the C2OH, but when you did that, this CHO is going to be where this hydrogen is, and the hydrogen is going to be where that CHO is. So they're non-superimposable. Okay, so a practice question. Which of these is the L enantiomer? Okay, so remember, think back to how you designate this. Okay, there's many carbons that are stereocenters that could have these D and L designations. But really, when we're using that term D or L for these sugars, we're talking about the hydroxyl group, or I should say the stereocarbon that is furthest from the carbonyl. So that's going to be, when we're looking at this, that's going to be this carbon. Okay, so those two carbons are where we need to look. Okay, and this is asking for the L enantiomer. So when we have the carbonyl on top of the Fischer projection, we look to the stereocarbon that is furthest from that, and then the OH group needs to be pointing to the left for the L enantiomer, just like in L-glycerolaldehyde. Okay, so that would be this guy. Okay, and this is the D enantiomer because OH is facing to the right. Okay, and you can also notice that if you drew a line there and acted like that was a mirror, these are mirror images of each other. Okay, so these are the same sugar. They would just have a different letter out in front of them, L or D. Okay. Some other types of isomers that you can have. The other term, you have an enantiomer for mirror image. Diastereomer is another type of isomer that are not mirror images. Okay. So diastereomers have different physical properties. Things like water solubility are going to be different for these. Okay. The example given here below are diastereomers of 4-carbon sugars. So these guys are going to be, they're not mirror images. So right, if you draw a line there, that would be a mirror image on that carbon, right? But this carbon is not a mirror image. Okay, so they're not mirror images, and they're not the same structure. So they're what we would term them as diastereomers. Okay, a type of diastereomer that we talk about in terms of sugars is epimer. Okay, and what an epimer means that it's a sugar or two sugars that differ only at one carbon, the configuration around one carbon atom. So if we look at the example given, D-glucose is shown here, and we'll look at glucose as one of the ones we'll look at quite a bit. Okay, glucose, we'll see this later, is an aldose. It's a 6-carbon aldose. Okay, so we have the aldehyde group at carbon 1. Okay, then and this is a structure that you'll have to know. We'll see it enough that hopefully you'll have a good idea of it. But really, how you have to remember it, if you're drawing the Fischer projection, okay, you start with the aldehyde carbon at 1, and then it goes right, left, right, right for the side that the OH group is on for D-glucose. Okay, and then carbon 6 is the non-stereo center CH2OH. So right, left, right, right. Okay. Mannose, you can see, has a different pattern. It's left, left, right, right. Okay, so these 3 carbons, carbon 3, 4, and 5, are the same, follow the same pattern as glucose. It's just carbon 2 is different. Instead of being right, the OH group on the right side, the OH group is now on the left side. Okay, so that's an example of an epimer, which an epimer is just a special kind of diastereomer. Okay, it's only differing at 1 carbon. Okay, another example of that is D-Galactose, that has the pattern of right, left, left, right. Okay, so that's, again, 1 carbon difference compared to glucose. So that carbon 4 now, instead of being on the right, the OH group is on the left. Okay, so mannose is an epimer at carbon 2. Galactose is an epimer at carbon 4. If you compared galactose and mannose now, those aren't epimers. They're differing between each other at 2 carbons, so that we would just term them as diastereomers. Okay, so let's try another practice question. Which of these hexoaldoses, so 6 carbon aldose sugars, are epimers of D-glucose? So let's look at our structure for glucose. It's given here, right? So we have right, left, right, right. Okay, so let's look at mannose. That's left, right. Excuse me, left, left, right, right. So that was 1 at carbon 2. Altrose is, if we look at altrose, it instead of right, it's left, the first carbon. Then it's also different at carbon 3, so that altrose is not an epimer of glucose. Differs at 2 carbons. Allose, however, is, right? So it's allose is, I mean, that one kind of easy because you can think of all the hydroxyl groups are all on the same side, right? So allose, we have all hydroxyl groups on the right side. Okay, so that would be an epimer at position C3. I should probably draw that a little bit bigger. Put a star there. Right. Gluose, we have right, right, left, right. So it differs at 2 carbons. Idose, we have left, right, left, right. So that's differing at 3 carbons. Galactose, it's right, left, left, right. So that is an epimer. It's an epimer at 4, carbon 4. And talose, we have left, 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 right. And so that differs at 2 carbons, so that is not an epimer. So the epimers, long story short, are allose, mannose, and galactose. Okay. So just a review of our stereochemistry. The 3 terms we need to be familiar with, first and foremost, that enantiomers. Those are the D and L, okay, forms of sugars, and those are non-superimposable mirror images. Okay. The next terms are sort of related. Okay, diastereomers is where you have an isomer that are not mirror image to one another. Okay, and epimers, that's a special type of diastereomer for sugars, where they're diastereomers only at 1 carbon. Okay. So I get a lot of questions when I teach this part of the course, like, what sugars do you need to know? Like, which ones do you have to memorize? And really, I think there are only about 4 that you really need to be able to, be able to draw or recognize and know the names of. And that's really because you see these a lot when you go on further in biochemistry, whether it's in metabolism or DNA, cofactors for certain enzymes, that sort of stuff. These are really important. Okay, so the first is glycerolaldehyde. Okay, that's the simplest sugar. It's 3 carbons long. Ribose is really what I would consider to be the standard 5 carbon sugar. Glucose is the standard 6-carbon sugar, and we've kind of already gone through that structure. And then finally, fructose is very similar to glucose, but it's the ketose form. So glucose is an aldose. Fructose is the ketose form of glucose. Okay, so let's go through some of these structures. Okay. And for this, we're looking at for aldoses, and in particular the D enantiomers of these. So for 3 carbons, okay, those are called trioses. For 3 carbons, really there's only one that in the aldose form. That's glycerolaldehyde. Okay. And this is, again, D enantiomer. And that's all based on the position of the OH group on this carbon. Okay, for 4 carbons, right, we have erythros and threose. And those are shown here, right? Those are both the D form because OH at the carbon, the stereocarbon that's furthest from the carbonyl, OH group is pointing to the right. Okay, so erythros, the OHs are on the same side, and threose, the OHs are on opposite side. These structures you don't need to know, though. Okay. The 5-carbon sugars, which are known as pentoses, okay, these are the 4 of them, at least the aldose forms of them. Okay, ribose is shown here, and ribose is one of those that you should know. Okay, luckily ribose is pretty easy. Okay, you have the aldehyde, carbon 1, and then carbon 2, 3, and 4, the stereocarbons, OH groups are all on the same side. And so for the D form, they're all on the right side. Okay, and then you have the CH2OH. Okay, this is not a stereocenter because this bond can rotate there, and this carbon is attached to only 3 different things, not 4, right? Because there's 2 hydrogens attached to it. So that's not a stereocarbon. Right. Okay, the other ones are shown here. Arabinose goes left, right, right. And xylose goes right, left, right. And Lyxose, never actually pronounced that one, but it's left, left, right. Okay. Again, the only one you really need to remember for these is ribose. Okay. And finally, the 6-carbon sugars, or the hexoses, okay, are shown here. Okay. Again, the one you really need to know is glucose. And again, that's right, left, right, right. Okay. The other that are pretty closely related to it, mannose and galactose, are shown here. Those you probably see a little bit more of in metabolism. They're both epimers of glucose, and those are used in the body a little bit more than some of these other sugars. Okay, so those are the aldoses. Now let's go through and do the same. Look at the same structures, but the ketose forms of these. Okay, so for 3 carbons, now we have, instead of an aldehyde group at the carbonyl at the end, we have a carbonyl in the middle, and that's the ketone functional group, so that's why we call these ketoses. Okay, so for 3 carbons, that would be dihydroxyacetone. Okay, 4 carbons, and you'll notice here there's no stereocenters, so there is no L and D form of that one. Okay. For 4 carbons now we've introduced 1 stereocarbon. Okay, so the D form OH groups on this side, the L form OH would be on the other side. Okay, and that's erythrulose. Don't really need to remember that one. 5 carbons, you have ribulose and xylulose. So very similar to the names ribose and xylose that we saw for the aldoses, just slightly different. Okay. 6 carbons, these are the ones, it contains one of the ones we'll need to know, and that's fructose. Okay, so fructose, again, it's the ketose form of glucose. So this is our carbon 1. This would normally be a carbonyl group in the aldose form for glucose, but now that carbonyl moves into what would be carbon 2, okay, forming the ketone. So then we lose a stereocarbon. So in glucose we had 4, and it went right, left, right, right, but now we lose that one because it's now the carbonyl, and we're left with left, right, right. So for fructose, you just have to remember left, right, right. At least in the D form of fructose. Okay, so that brings us into the how do I say this? So those are the structures, the open forms of these sugars. They can also form what are known as ring forms or closed forms of these sugars. Okay. And that's because of the functional groups that are contained within these sugars. Okay. So aldehydes and ketones, those carbons are electrophilic. Okay. And alcohol oxygen groups are nucleophilic. So you have nucleophiles and electrophiles in the same chain molecule. So what can happen is you can get an electrophilic attack or a nucleophilic attack on the aldehyde carbon or the ketone carbon by these oxygen groups. Okay. So when aldehydes are attacked by the alcohols, they form hemiacetals, and we'll show those structures here in a second. Okay, when ketones are attacked, they form hemiketals. Okay, so in this example here, we have an aldehyde. So we have this aldehyde functional group, and we just designate this as R1, right? Whatever the rest of this molecule is. And then the alcohol group here would be attached to something we call R2. So it's just something else, but it differs from this. Okay, so you can get a nucleophilic attack from this oxygen to that carbon, and when you do, you're going to form this sort of structure. So you have a carbon with an alcohol group on it. It's attached to one R group, one hydrogen, and then an OR group. What would that be? An ether. Right. So you have in the combination of this OH and that O attached to another R group and a hydrogen on this carbon is what we refer to as a hemiacetal. Okay, it's that's a hemiacetal. An acetal group is if we had replaced the alcohol group with another OR group, and this is what we would refer to as an acetal group. Okay. On the ketose sugars, what you can have is a nucleophilic attack from the oxygen to that ketone carbon. And what that forms is what's known as a hemiketal. Okay, so for hemiketals, again, you have a carbon attached to an OH group and an OR, but you also have two other R groups on the carbon. So you're replacing one of the hydrogens in the hemiacetal with another R group. Okay. And again, if you replace the OH group on that with another OR group, that's what we refer to as a ketal. Okay. So how do these cyclize? How do the rings form? Well, pentoses and hexoses, so 5 carbon sugars and 6 carbon sugars, readily undergo this intramolecular cyclization. So that means you have within the same molecule, a nucleophilic attack and a ring being formed in a ring or a loop, you can think of it being formed in one molecule. Okay, within the same molecule is what the intra stands for. Okay. The former carbonyl carbon becomes a new chiral center. So wherever that carbonyl group was that is being attacked by an OH group is now a new chiral center, and that's called an anomeric carbon. Okay, so that's important to remember. Anomeric carbon is the carbonyl carbon in the open form. So in the ring form, we call that the anomeric carbon. That is where the carbonyl group was in the open form. Okay. That former carbonyl oxygen is going to become a hydroxyl group, and that position can either be, if you think of a ring structure, that new OH group can either be above the ring or below the ring. And we term those two choices as alpha and beta. Okay, and how you can tell alpha and beta apart, you use the CH2OH moiety on the sugar. Okay, so if the hydroxyl group is on the opposite side of that CH2OH, and that will be the last sugar in the open form chain. Okay, so if it's on the if it's on the opposite side of that CH2OH group, you can think of that as being sort of like a trans isomer, thinking back to some terminology from organic chemistry, and that's the one we term as alpha anomer. If the hydroxyl group is on the same side, so you can think of that as being sort of like cis, then we term that as the beta anomer. All right, so let's look at this in glucose. So in glucose, the open form is shown here, right? And we have, for glucose, we can form a ring structure if we have a attack from the OH group on carbon 5 attacking the carbonyl group at carbon 1, right? That's going to make, let's see, 1, 2, 3, 4, 5, and 6 membered ring. Okay, the ring structure is going to have 5 carbons in it and 1 alcohol. Right, so this picture here is showing that open form structure just sort of bent a little bit. So this OH group is in closer proximity with carbon 1. Okay, so we have that attack, that nucleophilic attack by OH group on carbon 5. Okay, that forms a ring, and the oxygen then, the electrons, you can think of them if you're pushing electrons, right? These electrons go there, those electrons would come out and grab a hydrogen. This OH group that's formed can now either be above the plane of the ring or below the plane of the ring. Okay, so if it goes above the plane of the ring, that's on the same side as our CH2OH group at carbon 6, right? So that's what and you can think of it as kind of like a cis isomer, right? They're on the same side. That's what we determine as the beta anomer. And if they're on the opposite side, the OH is on the opposite side, that's kind of like a trans isomer. And that's what we refer to as the alpha anomer. Okay, so these ring structures we'd call be beta D-glucopyranose and alpha D-glucopyranose. Okay, that term pyranose is telling you it's in a ring structure. You can also do the same thing with fructose. So if we look at fructose's structure, shown here, that carbon the farthest carbon from the farthest, I should say, OH group from the carbonyl group can attack it. And what you'll notice here, right, for fructose, it's a 6-carbon sugar, but that carbonyl carbon is 1 carbon closer into that chain, 1 carbon further into the chain. So our ring that we're going to form is going to be 1 member shorter. And so drawing it in a little bit easier picture to see how this ring forms, right? This is our OH group on carbon 5. It attacks now carbon 2, not carbon 1, and we're going to get a 1, 2, 3, 4, 5 membered ring. Okay, 4 carbons and 1 oxygen. Okay, so that is shown here. Okay, when that happens, the OH group that's formed, right, can either be below the plane or above the plane. And so for this 5-member ring that forms, we get what's known as would be the alpha anomer, right? Because we're comparing it to the position of this CH2OH that's on this end of the molecule, furthest away from the carbonyl. Okay, so OH group is below the plane, this is above the plane, so this would be the alpha anomer. Okay, if that OH group is on the top and CH2OH is on the bottom, that would be the beta anomer. And that's, I want to point that out specifically for the 5-member ring structure for fructose and other sugars. Determining the anomer can be a little bit tricky because you're comparing the CH2OH group furthest from the carbonyl, that position, with OH, not with this other CH2OH from carbon 1. Okay, because this right here, if you look at these two, you would say, oh, they're on the same side. That's cis, and that should be beta. But remember, you're looking at this relative to the OH group. Okay, so that's, just wanted to point that out. Okay. Fructose is a little bit strange in that it can make a 6-membered ring as well. So these ketose forms of 6-carbon sugars, you can have a nucleophilic attack from the OH on carbon 5. You can also have a nucleophilic attack from the OH on carbon 6. Okay, so that would attack carbon 2 again, and now we have a 6-membered ring. Okay, when we do that, though, you can see that we don't have any CH2OH groups sticking up, so that would be bare. Okay, so when answering that anomer question, you're looking at the OH group in terms of where that CH2OH would have been. Okay, let me go back one slide. So when we're talking about glucose, the ring forms of glucose and the ring forms of fructose and ribose too, in a way, those are the structures I would expect you to know the open forms of, but it's also important to note when you draw the ring forms of them, are the OH groups going to be down, facing down, or up at each of these carbons? Okay, and so for that, you really have to walk through this and look at these and kind of convince yourself which direction they're going to go. Okay, or you can sort of memorize it too. So you can, starting from the anomeric carbon, right? That's going to differ in glucose. If it's alpha, it's down. If it's beta, it's up. But from carbon 2, 3, and 4, the OH groups would go down, up, down. Okay. And again, they alternate here in the straight line form, right? The right, left, right. So you're going to have alternating either ups and downs for the ring form. Okay. And with fructose, the 5-membered ring is a little bit easier, right? Because in 5-membered ring, you're looking at carbon 3 and 4, right? So they're going to be on opposite sides. For fructose, you have to remember it's up, down. And then when it's making the 6-membered ring, it'd be up, down, down, right? That's because now we're looking at carbon 3, 4, and 5, right? So 4 and 5 have the OH group on the same side. Right? A 5-membered ring form of a sugar is called a furanose. And where those come from are because they look similar to some organic structures. So 6-membered rings, sugars that contain an oxygen in the ring. So how do I say this? They're 6-membered rings. They have 5 carbons, 1 oxygen. It looks like pyran, the organic chemical pyran. That's why they term these as pyranoses, because it's similar to that structure. Okay. 5-membered rings that contain an oxygen look like furan, and that's why we call them furanoses. Just by convention, the anomeric carbon when we draw these ring forms is usually drawn on the right side. Okay. You can draw it on the left side, but then the whole molecule is sort of flipped around. So you really need to be careful if you're trying to draw these with the anomeric carbon on the left instead of the right. Okay, let's look at the structures of some of these. Okay, and these are structures I would expect you to know, hopefully be able to draw, although on an online exam, probably not going to ask you to draw them, but being able to recognize them would really help. Okay. Alpha D glucopyranose. Okay, there's a lot to unpack there. The first place we start is alpha. That means the anomeric carbon is in the alpha anomer, right? So we have CH2OH up and OH down. That's sort of quasi-trans, so that's the alpha anomer. Okay, D lets you know the position, which would have been on carbon 5. Okay, glucose means it's that 6-carbon sugar with this particular OH pattern. Okay, and pyranose means it's forming this 6-membered ring. Okay. Beta D-glucopyranose, the only difference we have here is that it's the beta anomer, so OH group is up in the quasi-cis isomer. Okay. Alpha D fructofuranose. Okay, that's fructose in a 5-membered ring. OH group is down. The furthest CH2OH group from the carbonyl is up. Okay, and then the beta anomer OH group would be up, and this the same side is the CH2OH on carbon 6. What else should I highlight? The pattern. Okay, again, the pattern for D-glucose in the pyranose form is going to be from carbon 2, OH is down, carbon 3, OH is up, carbon 4, OH is down. It follows the same pattern there for the fructose, the 5-membered ring form of fructose, right? But in this sense, we're looking at carbon 3 and 4, so the OH is going to be up on 3 and down on 4. Up on 3, down on 4. Okay. So those are monosaccharides. It gets a little bit more complex when you take 2 monosaccharides and stick them together to form a disaccharide. And you do that via what's known as a glycosidic bond. Okay, 2 sugar molecules are joined via a glycosidic bond between an anomeric carbon and a hydroxyl carbon. Okay, the glycosidic bond becomes what's known as an acetal, which we kind of showed that structure functional group earlier on when we were talking about hemiacetals. Okay. An acetal between monomers is going to be less reactive than a hemiacetal. Okay, so an acetal group we would term as non-reducing. A hemiacetal would be reducing. So when the term reducing sugar or non-reducing sugar is used, that's what that's talking about. A reducing sugar has a hemiacetal functional group. A non-reducing sugar has an acetal group. Okay. So any carbon that has its anomeric carbon involved in a glycosidic linkage, which forms an acetal, is going to be non-reducing. So a disaccharide formed upon condensation of two glucose molecules via 1,4 bond is called maltose, and we're going to see examples of that. So this, we have two glucose molecules. Okay. On the left, we have alpha D-glucose. On the right, we have beta D-glucose. A glycosidic bond can form between the anomeric carbon, so carbon 1, on one glucose molecule, and the OH group on carbon 4 from another glucose molecule. Okay, so when that bond forms, we get a release of water, shown here in pink, right? So this is what would be known as in this direction, a condensation reaction, because you're producing water. Okay, and what's left is you have a bond between the anomeric carbon 1 on the left glucose monomer and the oxygen, the OH group, on carbon 4 of the right glucose. Okay. How you write this out, you would term this as alpha D glucopyranosyl. Okay. Glucose meaning glucose, pyran meaning a 6-membered ring, and osyl meaning you have a glycosidic bond there. Okay. And then you would indicate what that bond is, between what carbon. So on this monomer, it's between carbon 1, and then you draw an arrow to the carbon it's attaching to on the next sugar, which would be carbon 4. Okay, and this sugar is D-glucopyranose. Okay. So ring forms exist in equilibrium with open chain forms. So you can have an example here of this disaccharide, and this would be maltose again. So we have our glycosidic bond between carbon 1 and carbon 4 on two glucose sugars. In this group here, remember, at this carbon now, we have an OR and another OR, a hydrogen, and an R group. So this would be an acetal functional group at this carbon. So this ring is not going to be able to open and close anymore. Okay, this is a fairly stable functional group. On this other end, we have carbon attached to OR, hydrogen, an R group, and then an OH. And this is what's known as a hemiacetal again. And so a hemiacetal is not as stable, and this will have some equilibrium between the closed form and the open chain form. So this will, open and close. Okay, when it does that, you'll get some sort of a mixture between when this closes again, you'll get some sort of a mixture between the alpha and beta anomers. So this OH group can change positions up and down. Okay. Why do they call this a reducing sugar? Right. So this would be called a reducing sugar because it has a hemiacetal, and this is a non-reducing sugar. Well, they call them reducing sugars because they can reduce the fact that they have in the open form an aldehyde means that aldehyde can reduce copper or silver in these different tests. Okay, in these tests, the historic tests using copper or silver really can tell you if you have reducing sugars in a sample. Okay. So that's what that term comes from. These sugars, because they have aldehydes in them, can reduce these metals in Fielding's test and Toland's test. Okay. Currently, those tests really aren't used anymore, or at least extensively. There's more modern ways of doing that, and one way is using coupling the reaction of an enzyme, glucose oxidase. Okay, when glucose or reducing sugars are around, it converts oxygen to hydrogen peroxide. Hydrogen peroxide then can change a certain coliphore molecule to alter its structure and change its color from a faint orange to a bright orange. So you're picking up a color change if the reducing sugar is around. Okay. Non-reducing, when the ones, at least the terms that we use for disaccharides in terms of a glycosidic bond between an anomeric carbon on one sugar and a OH group on another sugar, that will lead to a reducing sugar on the far end all the time. Okay, but you can also have a case where two sugar molecules can be joined by a glycosidic bond between two anomeric carbons. Okay, so the anomeric carbon on one sugar can form a glycosidic bond to the anomeric carbon on another sugar. Okay, when that happens, you get a disaccharide that has no reducing carbons on it. Okay, so the product formed will have two acetal groups and no hemiacetals. And because of that, right, the rings on these sugars aren't in equilibrium with the open forms. Okay, there's no aldehydes around, and they're non-reducing sugars. So if we look at some examples of different disaccharides, the first example up here would be lactose, and I'm sure you've all heard of lactose. Okay, what lactose is it's a disaccharide between galactose and glucose. Okay, so galactose is in the beta anomer form, so the OH group would normally be up, same side as the CH2OH. Okay, and it's forming a glycosidic bond between that carbon 1 in galactose, and carbon 4 in glucose. So this is the shorthand way of writing this disaccharide would be gal for galactose, beta 1 to 4 glucose, GLC. Okay. And you'll notice this bond here is we draw this glycosidic bond a little bit funky. Okay, this is shown as like a, I don't know, kind of a curve, an oxygen, and then a curve down here on the other side. And then if you have the other way of drawing it is, they're on both the same side. It kind of curves around and goes down. Okay, this is just an easy way to show so these sugars remain in the sort of same orientation that we're used to drawing them. Okay, because if you really actually drew this how it is three-dimensionally, this this sugar here would be sort of flipped, either upside down or sideways, and it would just be a little bit harder to look at. Okay, so that's why these are drawn a little bit funky, the glycosidic bonds. It's to keep the carbons in the same relative orientation to one another. But they don't actually show the exact structure. Okay. Another disaccharide that you've probably heard of is sucrose. And what sucrose is it's a disaccharide between fructose and glucose. So it's fructose, and this is a little bit funny. So this was fructose and this is glucose. Fructose and also sucrose is a non-reducing disaccharide because it has the glycosidic bond between two anomeric carbons. So here we have fructose. Fructose looks a little bit different in this case because it's drawn upside down in a sense, because we're used to drawing the anomeric carbon on the right side, but in this case we have to draw it on the left side because it's forming a glycosidic bond with the anomeric carbon of glucose. Okay, so when we do that, we're flipping it around and it looks sort of upside down, okay, to what its typical orientation would be. Okay. And that glycosidic bond is between the beta 2 anomeric carbon on fructose to alpha 1 carbon on glucose. And finally, trehalose is a non-reducing disaccharide between two monomers of glucose. Okay, and they're both an alpha 1 to alpha 1 glycosidic bond. Okay. So we have in the first case with lactose, we do have at carbon 1 on glucose. Here, the anomeric carbon, we have hemiacetal, right? So OR, R, OH, and an H. Okay, so that's a hemiacetal. So that is a reducing sugar. These other two sugars are non-reducing. Okay, so for sucrose, we have, if we're looking at the beta 2 carbon, what's that attached to? It's attached to OR, an R group, an O group, an OR group, and another R group. So 2 R groups and 2 OR groups. So that would be a ketal form, right? That's non-reducing. And then finally, trehalose at the glucose at carbon 1 would be OR, OR, R, and H, which is an acetal group. Okay, again, non-reducing. Okay, so next time we'll continue on in chapter 7 and get through section 2, talking about polysaccharides. Okay, these are sugars that are attached in longer chains, so they get a little bit more complex, but they still follow the same sort of pattern of this glycosidic linkage. Okay, and finally, section 7.3 talking about glycoconjugates. So when sugars attach to other biomolecules like proteins.